central dogma of molecular biology proposed by F.H.C. Crick. What is this central dogma? The flow of genetic information. He named it as central dogma. The flow of genetic information. Crick named as central dogma. According to this, the genetic flow is unidirectional or linear. According to him, from DNA is formed by autocatalysis. From DNA, by transcription. mRNA is formed by translation from mRNA. Protein is formed. That protein gives the character of that organism. So, based on the information present on DNA, particular protein synthesizes. That protein gives the character of that organism. That produces the structure of the body or structure of that organism, that part of that organism. So, character of an organism depend on information present on the DNA. So, the flow of genetic information from DNA to protein, according to Crick, it is the unidirectional or linear. But in retroviruses, those viruses with RNA as genetic material called retroviruses, in them, this direction is reverse direction. It is the forward direction and reverse direction of genetic flow observed in retroviruses. Example, HIV. The genetic material in HIV is the RNA. This RNA that produce DNA. Again, the DNA produced RNA. So, formation of the DNA from the RNA, reverse transcription. Formation of the RNA from the DNA, transcription. And the formation of the DNA from RNA, reverse transcription, and the enzyme reverse transcriptase. So, it is reverse direction. The flow of genetic information is reverse direction in retrovirus. But in most of the organism, the flow of genetic information is forward direction.